Hello everyone, this is Mr. Jackson, and we're going to be moving forward with Lesson 8, going into Part 1b, where we're still looking at the work of Gregor Mendel, but now we're going to be specifically applying his findings, specifically over the principle of segregation, into one-factor, or what we call monohybrid crosses. So let's get right into it. You'll know that you've mastered this portion of the lesson when you're able to explain how segregation, specifically the separation of the different alleles from mom and from dad, can actually be shown using what we call a Punnett square. And then you're going to be able to use that Punnett square diagram to predict the genotype and phenotype combinations from these genetic crosses. And if this sounds like a whole lot of jargon, it might just be because these words aren't all that familiar to you yet, and we'll take care of that here very, very soon. So really quick, want to review Mendel's experiments, some of the key points here. So in these experiments, he made sure of three things. The first thing is that he had full control over how these plants reproduced. And he did that by choosing to cross-pollinate specific plants with each other. So he prevented self-pollination, which would typically happen, and chose which traits to cross specifically. He used only purebred plants, meaning that each of these organisms had ancestors that were genetically uniform. So there wasn't any sort of interference from previous generations having variety of traits. These were self-pollinators, so typically their offspring are going to be identical to parents, generation after generation. And so he kept these really purebred plants for these experiments. And then finally, he observed very simple either-or traits. So this is 7 traits that he looked at in these pea plants, and there were only 2 possible versions that those traits could come in. So like green seeds or yellow seeds, tall plants or short plants. And this is a lot simpler than a lot of traits that exist. Easy example would be for human beings an eye color, right? It could be a whole lot of, different versions. It could be like blue, green, brown, but also it kind of exists on a spectrum where there's a whole lot of variety of those in-between colors. And so it's not always as simple as this or that, 1 or 2, but in these very, very early stages, it was extremely useful to keep those things simple. And want to make sure we understand, whenever we're referring to a cross, we're just talking about the mating of 2 organisms. So if I say a one-factor cross, I'm looking at a specific trait, one trait that happens whenever we mate 2 organisms. In this case, we're using pea plants. And if you see the letter P, that just represents the initial or parent generation that we're looking at. And anything after that, F1, F2, again, this is referring to filial, son or daughter generation. It's just talking about offspring. If it's F1, that's the first generation, F2, second, so on and so forth. And we want to make sure that we fully understand the significance of these alleles, these different versions that these genes can exist as, or these traits can exist as. Now we're going to represent these various alleles with different letters, specifically uppercase or lowercase letters. Uppercase letters are going to be used to represent dominant alleles, while lowercase letters are going to be used for the weaker recessive alleles. And so we'll expect to see the dominant alleles, those capital letters, a lot more often than the recessive alleles, which are going to be in lowercase and weaker. We have to remember, everyone has 2 alleles, one from mom and one from dad, for every single gene in our bodies. And whenever those 2 alleles come together, there's 2 main ways in which they can do so. We could have a homozygous combination, which means that we happen to get the same allele from both parents for a certain trait. So if I get both dominant alleles, one from mom, one from dad, then I would say that I am homozygous dominant, and that's what we would represent with 2 capital letter A's. If I happen to get both recessive versions, one from mom and one from dad, then I would have homozygous recessive as my combination. And so there's homozygous, meaning same version in the zygote, right? So they're the same. But then we also have heterozygous, which is different versions in the zygote. And so we could have two different alleles if I were to receive one dominant from one parent and a recessive allele or version from the other parent. And so if we're looking down here, this is all of the different combinations that could have come together from each of the parents, or really just from a self-pollination. If you think back to part 1A, where we had a male and a female portion from the same plant. And so whenever those came together, there were 4 different ways in which those could potentially combine. And we can see the versions and how those would actually come together here. And so you can see here, this would be that I got 2 dominant alleles. I have a dominant and a recessive in a couple of different options here. And then I finish with 2 recessives that would come together as well. And we want to make sure that we fully understand this principle of dominance, which is to say that a dominant or strong allele will always express itself over a recessive or weak allele. And so whenever I'm talking about dominant, I'm talking about the fact that if it's present, that allele is going to always have that trait expressed. So we'll always see the trait if we have at least one dominant allele. Good example is if we say that brown hair is dominant over blonde hair, we represent that with a uppercase letter B for brown and a lowercase letter b for blonde, right? So this shows me dominant brown and recessive blonde. And if I get even one uppercase B, one dominant version from either mom or dad, then I'm going to have brown hair. And so I'm going to have brown hair whether I get 2 dominants or even just 1. And that's shown here. Now, whenever it comes to recessive alleles, this typically, these traits are only going to show as recessive if we happen to get both recessive alleles and there's not a dominant allele in the mix. So an example here would be the only way that I could get this recessive trait of blonde hair is I have to inherit the recessive allele from both mom and dad so that my combination is specifically little b for blonde hair. And so I've been using the word combination, right? So combination of alleles, but there's actually more specific terms that we're going to use moving forward to mean combinations of these alleles. And that's genotype. And so a genotype is the actual combination of alleles that we inherit. So if we look to our image at the right, an example here would be the genes within the cells of this flower that actually code for flower color. And so again, this could be 3 ways in which this simple trait could come together. With I might have homozygous dominant, where I got 2 dominant versions. Heterozygous, where it's 1 of each, or homozygous recessive, where now I'll expect to see the recessive form of the trait. And then I have phenotype. Phenotype is the physical traits or characteristics that I see in organisms. So a good example here is what you can actually physically see for this flower, which would be purple in color. So again, genotype is the actual genes, right? The combination of alleles, whereas phenotype is the physical characteristics that you can see. And we want to make sure that we fully understand this principle of segregation, where we have chromosomes that will actually separate during meiosis, and in doing so, each gamete, that's sperm if you're in males, eggs if you're in females, they're going to end up receiving only 1 chromosome from each pair. An example is neatly laid out here, right, where we have a man that has alleles for brown hair and blonde hair. So he is heterozygous. He has both versions in his genotype. So he is heterozygous for hair color. This happens to mean that his sperm cells will contain the allele for brown hair or the allele for blonde hair. It's going to be one or the other. He's not going to pass along both because they're going to segregate from each other or separate during meiosis. And so each of his 4 gametes that he creates will ultimately only get one of these alleles, either for brown hair that he could pass to his offspring or blonde hair that he could pass to his offspring. And so now we get to this guided notes portion of our part 1B, where I'm going to be handing out some physical copies, some papers that you can work through. I highly encourage you to follow along and work through those along with me. So I'm going to use my pen tool here, and basically we're going to break down how we can use these diagrams called Punnett squares in order to represent all of the possible combinations in our offspring whenever we combine parents that have different combinations or different genotypes and we cross them. So I'm going to hop over here and grab my pen tool, and we're going to work through this. So whenever I have a Punnett square and it's represented here with 4 boxes, I'm going to call this a monohybrid or one-factor cross. Mono referring to one, hybrid meaning that I'm taking parents that are different to create hybrid offspring, or one factor meaning that I'm only looking at the potential outcomes for one specific trait. Okay, so this is a one factor or monohybrid cross between 2 organisms, still just looking at one trait. So I'm going to take the known genotypes of each parent for this trait. I can see for parent 1 that I am heterozygous dominant because I have one allele that is dominant and another allele in this genotype that is recessive. I see that for parent number 2 on the left-hand side, I have the same thing, right? It's heterozygous. They're different. I've got a dominant capital A and a recessive version that I could pass along as well. And so in order to know how these different alleles will segregate and could come back together during fertilization, all I have to do is drop the alleles on the top down into both of their columns and the alleles on the left over into each of their rows. So I can show you how this works here, where I might start with this left column where I drop this uppercase letter A down into both of these boxes that come below it. I can hop over to this recessive version. This little letter A will go into each of its boxes that are down and below. And then I can go over to the left and show how parent 2 could pass along each of their alleles, right? So that uppercase A, that dominant version, goes into each box in its row. And then finally, this recessive allele is going to go into each box in its row. And by doing this, this is showing me 4 potential genotypes that could make its way into my offspring for a certain trait. And if I know what the genotype combination is and what it represents in terms of a trait, I can also tell you what the phenotype likelihood is as well. And so in this last slide for part 1B, I'm going to walk through a few examples for how to take these unique scenarios and to put them into a one factor or monohybrid cross. And then using that information that I solve for, I can say, what is my likelihood for having certain genotypes in my offspring, as well as the physical forms or the phenotypes that those can take. So let's work through a few examples. And again, this will be what you'll follow along with in your guided notes. So please do feel free to follow along. But in the first one, it says, in pea plants, round seeds are dominant. And so for this, I'm just going to choose, for round, I'm just going to use the uppercase letter R. So I can say that round seeds are uppercase R because they're dominant. Wrinkled seeds are just going to be lowercase because they're recessive. So it tells me to cross a homozygous, so they're both going to be the same, and recessive. So homozygous recessive. So that's for parent 1. I'm going to use 2 little letter R's here. And I'm crossing that with a homozygous dominant. So it's going to be dominant, and they're both the same, meaning that I know that they're the same and dominant. Both are uppercase letter R's. And so to solve this, to see what my potential offspring could have, all I'm going to do is just basically drop each of these little letter R's into their columns. So that's the first allele for parent 1, and then the second allele for parent 1. I'll go over to parent 2. Remember, this is going to come across to each box in this row, as is the second version down in the bottom row. And by doing so, you can see that no matter which way my gametes from parent 1 and gametes from parent 2 could come together, I'm always going to receive the same outcome. So there is no doubt in my mind what the genotype of an offspring from this cross is going to look like, right? So I can go over here to my genotype ratio, and for my genotype ratio, I can say that, well, 100% of the time, I'm going to have a genotype that is big R, little r. And that's because every single box where it shows those potential outcomes, that's exactly what it shows. And so for my genotype ratio, I represent ratio with 1, big R, little r. And for every one of those that I have, I can expect to also see 0 of anything else. Okay? So if I have a big R, little r, that means there's nothing else it could possibly be. And for my phenotype ratio, I know that if I have at least one big R, that this is going to, whoops, 1 second there. See if I can exit my drawing here. Sorry, it's going to erase everything, but hopefully you had that written down. So I'm going to come back here. So I got my phenotype ratio, and whenever I have my phenotype ratio, what this is going to do is I know that all of those boxes had big R, little r. If I have at least one big R in my genotypes, I know that it's going to mean that I have round seeds. So I could say 100% of my phenotypes are going to be round. And if I'm setting that up in a ratio, again, this is going to mean 1 round seed, and the other options that were available at the time were 0. There was nothing else that it could possibly be. And so my phenotype ratio was 1 to 0, and as we remember, hopefully we saw the genotype ratio was 1 to 0. So let's hop down to our second example here. Now it tells me to cross 2 heterozygous plants. So let's work through this as well. So if I know that they're 2 heterozygous, I know that the first letter is going to be a big R, and the second one is going to be different. It's going to be a little r. So I've got 1 dominant and 1 recessive, and that's going to be for both of the parents that I'm crossing. I do the same thing like normal. I'm going to drop down my big R from the top into both boxes in its column first. This little r drops down into both boxes below it. And then I move this, again, this big R over into both boxes in its row. And the little r comes over as well. And so now I've got something different from the first one, where I have some different options, some different things that this could actually become. And so I'll set it up in percentages first, just to keep it easy on myself, where I can see that 1 out of the 4 boxes, or 25%, ended up with big R, that upper left-hand box. I see 2 boxes, or 50%, so half of them came together to give me big R, little r. And again, I had 1 out of 4, or 25% of the time, I had homozygous recessive as an option, and that was little r, little r. Now again, if I'm going to represent these percentages as a ratio, I know that it's out of 4. So if I have 1 box that could be big R, that will be a ratio with 2 boxes that could be big R, little r, and 1 final box that could be 2 little r's. So I've got 1 dominant homozygous, 2 heterozygous, and 1 recessive homozygous. And then in terms of phenotype ratio, again, it's going to be, I'm looking at the different ways in which the physical form could appear. I know that big R, and big R, little r both result in round seeds. And so since there are 25% and 50%, I know that 75%, right? 3 out of those 4 boxes can result in round seeds. And then that leaves 25%, which will be the other option. And in this case, we'll know that that 25% will be wrinkled. Pen tool's lagging just a little bit, so doing the best I can with the font here, guys. Sorry about that. And then finally, so now that I have these potential phenotypes, the physical forms, right? So I've got both of my rounds represented as well as my wrinkleds. Then now I can see that for every 3 boxes where I have rounds, I'm left with 1 box that would end up maybe being wrinkled. So I've got 3 dominants with 1 recessive phenotype. Let's go ahead and finish up here with our last example where it tells us to cross a heterozygous plant with a plant that has wrinkled seeds. So if it's heterozygous, again, I know that it's going to have 1 of each. They are going to be different. And it tells me with wrinkled seeds. Okay, so it didn't tell me exactly the genotype, homo or heterozygous or recessive or dominant, but I know from what it told me earlier that wrinkled seeds are recessive. And if I have wrinkled seeds at all, the only way in which I can have those wrinkled seeds is if both alleles are recessive. There can't be a dominant one here and expect to see a round or a wrinkled seed. So I'm going to do exactly the same method, right? Where I go to my column, I drop that big R into both boxes. I go to my second column and I drop that little r down as well. I go to my first row, I bring that little r over. Same thing on the bottom row, bring that little r over. And now I have my 4 potential offspring, what those genotypes might be. I can see that 2 of them are the same on both columns. And so I could say that my genotype ratio is 50%, where I have big R, little r. So I've got 50% heterozygous dominant, and I've got 50% or half where I'm homozygous recessive. And so again, I could say that for every 1 big R, little r, I can expect to see 1 little r. So for every 1 heterozygous, I have 1 homozygous recessive. And in terms of phenotype ratio, I've got half of these that would actually give me round seeds because I have 1 dominant allele. So again, I've got 50%. And that's 50% round. And also I have 50% for the recessive form, which is 50% wrinkled. And because we're still working, guys, with 50-50 here, take a second to finish this word. Because I'm still working with 50-50, I'm still working with 1-to-1 ratio. Because for every 1 round that I might have, I also have 1 wrinkled that it could be as well. And so that's going to actually conclude our demonstration and our guided notes portion of the Part 1B for Lesson 8. And you can feel free to go back and to watch how we solve this as many times as you need to. Please utilize those guided notes. I think they'll help a lot. And we're going to be working on this a lot in our practice activities, again, with that handout packet that I'm going to be giving you all in class. And so I'm going to stop it here. I know this has been rather lengthy, but a very, very important part to the lesson. And I hope that you found this useful, and we'll see you at the next one. Thanks, guys.